So we're going to be talking about the Calvin cycle now, or the dark reaction of photosynthesis. And keep in mind that the Calvin cycle's purpose is to take that carbon dioxide that the plant has breathed in through the stomata and fix that into a sugar molecule. And I mean fix it in the terms of it's incorporated into making a sugar. So this process I'm going to show you by this short movie is going in more detail than you need, but it definitely shows you where the ATP and NADPA are used, and it shows you how those carbon molecules change over time. So I'll go ahead and play that. Keep in mind you do not need to know the names of each of these different sugars that are happening and the process, but you do need to know which step needs ATP, where that 3-carbon sugar goes to, and we'll go through that in the lecture afterwards.
So far, energy from light has been stored in the chemical bonds of ATP and NADPH. In the Calvin cycle, this stored energy is used to produce sugar molecules. The Calvin cycle is a complex series of chemical reactions carried out in the stroma. The Calvin cycle begins with the input of CO2. 3 molecules of carbon dioxide are added to 3 molecules of a 5-carbon sugar, abbreviated RuBP. These molecules are then rearranged to form 6 molecules called 3-PGA, which have 3 carbons each. Note that molecules are not actually rearranged in the manner shown here. In the next 2 reactions, products from the light reactions are used to boost the energy of these 3 carbon molecules. First, high-energy phosphate groups are added. 6 ATP molecules contribute phosphate groups, so that each 3-carbon molecule receives an additional phosphate group. Electrons are added in the next reaction. 6 molecules of NADPH are oxidized. The electrons from NADPH reduce the 6 3-carbon compounds, creating 6 high-energy G3P molecules. G3P, or glyceraldehyde 3-phosphate, is a sugar, the final product of the Calvin cycle.
One of the G3P molecules represents the 3-carbon dioxide molecules fixed so far. The other 5 G3Ps are reshuffled to regenerate the original RuBP molecules. To summarize, so far the Calvin cycle has used the energy of the light reactions to reduce 3 molecules of carbon dioxide and produce 1 molecule of G3P.
3 more carbon dioxide molecules are fixed to form G3P in this same complicated way. To make each G3P, the Calvin cycle consumes 9 ATP molecules and 6 NADPH molecules. These are regenerated in the light reactions. G3P is the actual final product of the Calvin cycle. The cell can combine 2 G3Ps to make glucose, which stores the energy that chlorophyll originally captured from the sun.
All right, so let's take a look at how this Calvin cycle happens step by step, and let me explain what things in fact you need to know, because you definitely don't need to know the terms. So just one more time, I'll go back one step. That the Calvin cycle uses ATP and NADPH that happened in the light-dependent reactions, and we went through that, and it makes its sugars, and the sugars that we're going to be dealing with is glucose. Okay, so let's see if I can get my pen to work here and get my thing. So the sugars, we're talking about glucose, and just keep in mind this glucose, the chemical formula is C6H12O6. All right, so let's take a look at how that happens. So first of all, you have the carbon dioxide that came through the stroma entering the cycle, okay? And excuse me, it came in through the stomata into the leaf, and then from the leaf, inside the leaf, it's gone through diffusion into the plant cell, and through diffusion this carbon dioxide moves into the chloroplast where it is now in the stroma. Okay, so this carbon dioxide is in the stroma of the chloroplast. So it originally came from outside of the leaf and then moved in. So when this carbon dioxide is in the stroma, there are proteins, and what these proteins do is they take that carbon dioxide and do something that is called carbon fixation. And simply that means that the carbon is now fixed into a molecule, so it's not by itself anymore, okay? And it does that through, again, proteins that will make this reaction happen, okay? So it's attached on this carbon dioxide to now a 5-carbon sugar. You notice it's a 5-carbon sugar because we have 5 carbons here. It now becomes a 6-carbon sugar. All right, we have 1, 2, 3, 4, 5, 6. All right, our next step is that 6-carbon sugar, with the help of a protein and with the help of ATP and NADPH, becomes a 3-carbon sugar. And there are 2 of them. I'm going to draw the second one here because you can't see those, okay? So there are 2 of those. And it requires ATP, which remember is that stored, it's the battery that's fully charged, and NADPH, which is also a battery that's fully charged. That protein uses that energy from these fully charged batteries to help break it apart and reshuffle things. So the next step is with the help of a protein. You have one of those 3-carbon sugars leaving into the cytoplasm. So it goes out of the chloroplast and into the cytoplasm of the cell. And that's where there are going to be other proteins that are going to wait for the second one. The other 3-carbon sugar stays in the stroma, okay? So that one stays in the stroma, and that's going to be part of the molecule of this 5-carbon molecule that will be used again to help the fixation of the next carbon dioxide. And in order for this to combine, you need to have 6 ATP. Usually building things into larger groups requires energy, and it's certainly the case here. So you have then ATP being used here. And then the cycle happens again, okay? So just to go, and the cycle actually keeps on happening. We'll just quickly do a review. We have carbon dioxide. Here we go. We'll have the notations. Carbon dioxide coming in through the stomata of the leaf through diffusion, then moving through diffusion into the cell, going through diffusion from the cell deeper into the chloroplast, and then when it's in the chloroplast, it's in the stroma, where it then attaches with the help of a protein to a 5-carbon sugar, which is right over here. That's called carbon fixation. So it becomes a 6-carbon sugar with the help of ATP and NADPH. This 6-carbon sugar is broken into 2 3-carbon sugars. Okay, so now there are 2 of those. One of those stays in the stroma to become the next 5-carbon sugar, and the other one leaves into the cytoplasm. All right, so there we can see that notation. And that 3-carbon sugar needs to become a 5-carbon sugar, and it does that with the help of ATP. And you're probably asking where these carbons come from. These molecules kind of always are broken apart and then put together again, broken apart, just like Lego pieces. And they always stay in the stroma, okay? And then in the cytoplasm, the last step is that you have 2 3-carbon sugars. There are my 2 3-carbon sugars. And with the help of a protein, they are combined to be 6 carbon sugars. And this 6-carbon sugar is C6. That's why we have that 6, H12O6. And that is the carbon cycle. The Calvin cycle. The only thing you need to know for the Calvin cycle is the general flow of it and what is being used. The idea that ATP and NADPH are used, and both of those things regenerated are fully charged as batteries in the light cycle.